We got Sean. Sean, you're in again, are you?
Hello, hello. Can't keep me
Oh,
away.
that's awesome. God, I wouldn't want to listen to me this many times. All right. Do we have everyone? Are we ready to go?
Can you see the screen or no?
I can.
Okay.
I just want to make sure we got everyone, then I'll kick it off.
Yeah, I think we're good to go.
Okay. All right. Well, welcome to the second version of Electrophysiology Bootcamp. And the focused goal of this version is to start to train your brains, which continue to this day to be the top pattern recognizers on the planet. And computers, as much as AI and computer science has tried, they have not been able to convincingly even say that they can match us in terms of human pattern recognition yet. And I hope that yet remains. So the point here is similar to when a radiologist looks at an MRI or a CT image. You can take yourself from when you look at these results to just looking like squiggles to being able to actually recognize the patterns embedded within them. And that's kind of what today's boot camp's all about. So we'll go to the next slide, please. All right, so we left off on last boot camp really talking about the fact that from the EEG, we do signal averaging to extract evoked responses. And critical to neurocatch is to understand evoked responses mean you need to stimulate the brain. You can kind of think about it like you stimulate and look for a response, similar to like a knee-jerk reflex sort of thing, except at a neural level. So this slide is dedicated to helping you understand how we stimulate the brain. Keeping in mind that there are lots and lots of labs around the world that have found all sorts of different ways. We condensed what would take 2 to 3 hours in a laboratory with the top paradigms that allow you to get the three components that you know, the N1, the P3, and the N4. So the left side, let's start with the top part of the graph first. So the top part of the graph basically shows you that what we did is we merged tones and word stimuli. So as everyone on this call probably knows, I think Lisa, you might not yet because you haven't got your neurocatch and you haven't listened, but you hear kind of a series of tones that go beep, 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 and then you hear a word pair. And the word pairs either match or don't match. And the key thing is that we do that more than once. We do that many times over the duration of 5 minutes. And so that gives us EEG recordings to each of those stimuli, the tones, the auditory tones, and these word pairs. So the bottom of this slide gives you kind of a diagram or a schematic of what we're doing with the tones. So the classic and very well studied oddball paradigm, which is what it's called in the literature, was discovered that if you present a series of tones, let's say at a certain decibel loudness, like 100 hertz or 1,000 hertz, 100, pardon me, then your brain gets ready and just listening to those. And if in an improbable or rare tone comes along that's louder, so all we do is 10% of the time put in these red tones at 120 hertz or decibels, then what happens is you get an oddball. So that actually is how we elicit both the N100 and the P300. So we can get the N100 to any of these tones, but when we actually increase the loudness, then we get a larger N100. So if we look at the red tones themselves, when it's much like when you hear, if you're in the woods and you're walking and a stick snaps and you pay attention. So you hear this louder tone and it creates a louder N100, but it also creates this P300 because it's rare and improbable. And so both the N100 and the P300 are come off from the tones. And there's in general, you need anywhere from 15 trials to upwards to get these, 15, 20. We get a sufficient number, but the name of the game is to get them off the kind of minimum amount to shorten the time. So in a research lab, you can imagine if you want a really good N100 and P300, you would just do it over and over and over and over and over again because you have the expensive time, and then you get really, really crisp waveforms. What we've been able to do is get them off of the minimum amount of time with a minimum number of tones and then use a lot of signal processing to extract them in a reliable way. So what's interesting is, certainly being Canadian and growing up and listening to, I would always remember the beep, beep, beep for NRC time and NRC standard time, and then you'd wait for it, right? And we figured out to merge the tones with the words to build an expectancy that you would wait for these word pairs. And these word pairs are really well studied. Basically, it's around the concept that if I give you something that you don't expect in speech or reading or otherwise, you will map the mismatch in the N400. So if I give you a matching word pair like bread, butter, now you hear the beep, 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 and you're ready to hear a word pair, you know it's coming, and then you get bread, butter, and that's what we would call a congruent trial. So that matches your expectation. And if we violate that, it turns out, and we say something like bread table or bread horse, that's an incongruent matching. And what happens then is you get this N400 response, because your brain catches that and says, hang on, that didn't fit with what I expected to hear and what was logical and matched. And your semantic processing, your cognitive processing kicks in and processes that. So what we do with our word pairs is you get the tones to get you ready, then you get a match or mismatch word pair. They're 50%, so 50 match and 50 don't. And there's one really cool trick embedded in this, is that it's built to be similar to if you land in the emergency room, for example, with a concussion, the first thing that their care providers will ask you is, do you know what day it is? Or do you know where you are? Basically, are you aware of your context? And half of these word pairs actually also test that. So they'll say something to the effect of Thursday, date, or place, and if you're in Vancouver, Vancouver. So the design is built to be smart and also test your awareness of matching context. When you get the N400, they're averaged together, but just FYI, we actually know how to pull that out and look at it. And we've been able to demonstrate that the context aspects are particularly sensitive to things like dementia. Next slide, please. Okay, so now we're into the nitty gritty. You've seen these slides before for the most part. Lisa, you probably haven't. These might be new to you, but when you get onboarded, I show you these slides. So the purpose of re-showing you these slides is to train your brain to find the components. So what I'm showing you now is that when we look at the N1 and P3, which you can see now come to the tones, that's why they're together. And we look at the N4, which comes to the word pairs, which is why they're separate. And we look at them across a group of both healthy individuals that are early adulthood and individuals that are further along in their adulthood. The responses are very stable. They're stable across lifespan, but they do actually demonstrate, if you look, you've got this N1 P3 complex, right? It actually has a larger complex embedded in it. So let's orient you to the graphs. First, this is a standard graph that you would present ERPs on. So on the horizontal axis is time in milliseconds. So what we're looking at is the brain wave over from 0, which is the dotted line, to 1,000 milliseconds or a second. So these are occurring very rapidly. And one of the things you can see is that on the vertical axis, this is the size of the response and also the polarity, like I talked about last time. And that's measured in microvolts. So we're looking at the 0 point in time and the 0 point in microvolts. And the dotted line, of course, shows a 0 point in time. So one of the interesting things that I taught you last time is that signal averaging averages the EEG noise to 0. And a way that you can check that is we actually start recording a little bit before the stimulus. It's called a pre-stimulus baseline. So you can see in these graphs, it goes to negative 1.1 seconds. And that demonstrates there that the lines are completely flat. So this is actually demonstrating that by signal averaging, you're canceling the noise and you have flat lines. So your pre-stimulus baseline is used as a way to check about the quality of your waveforms. So you can tell with these that they're very flat and it's high quality results. Now, if you go forward from the 0 point in time, ahead of the N100, you'll notice there's actually a little dip. Now, how do we name these things? I mentioned this on the onboarding training, but basically, if it's a negative going peak around 100 milliseconds, doesn't have to be exact, then it gets the name N100. So ahead of the N100, you see this positive, small positive going peak, which is also occurs around 100, and that's called the P1. So there's this complex for any stimulus that shows the P1, N1, and then often the P2 and then P3. And often you'll hear us talk either with ones or 100s, and they're interchangeable. So quite commonly, if you're first introducing a component to someone, you'll say N100, so you can teach them it's negative going in 100 milliseconds. But then as you get into shortening it, the field just goes to N1P2 or N1P3. And so what you're looking at there is an N100 and a P300. But you can actually see this complex of the P1 ahead of it, and then it actually skips right past the P200 and goes to the P300. And you can see that in the neurocatch sequence, that's pretty stable in both the younger and older adults. Now I want you to keep training your brain on that, because that's a really signature. That's almost like the QRS wave, right? It's a very, very signature waveform that you can use to check quality. And to make that point, take a look at it's not just for discrete tones, but when we play speech words, you see this P1, N1, P3 complex. And then after that comes the N400 in the word stimuli. So your eye can train and go back and forth, and you can see that, see the baselines are flat, see the P1 coming around 100 milliseconds, the N1 right after, and then it goes into a combination of what is a P2, P3. Now, why I keep saying a P2, P3 is because one of the problems with neurocatch right now that the gang is fixed is that the stimulus actually does in some people elicit a P200, and in others it elicits a P300. And that can create a problem with the peak picker. And we'll talk a little bit more about that, but I just want to introduce that concept. And then the last thing on the slide I want to train your brain into is look at the waveform shape. So we've been analyzing them in terms of what direction is the peak going, how big is the peak, when is the peak. But if you look at the N400 and you compare it to the N1 or the P3, you can see it's a lot broader. Now you'll be asking yourself why. And intuitively, the reason is that it's further in time and it's representing a lot more processes. So that early peak that you see is actually coding phonologically that the sound you expected to hear didn't fit. And then later, the peak where you see the arrow for the N400 is coding that the actual meaning doesn't fit. So we process and begin processing words about 200 milliseconds after we start to hear them. And so the reason the N400 has a broader peak and it's not as big an amplitude is because it's actually capturing a lot of complex cognitive processing. So Steph, if you can switch to the next slide now, you get to switch and see them again, but this is for an individual. So this is for like a single patient you might see. Now here, what you can see is that the first thing you'll notice is that they continue to be representative of what you saw at the group level. So again, take your eye through, right? Start in the top left-hand corner, look at the P1 and then the N1, and look at the P3. Look at the difference between the standard and deviant tones. So I showed you earlier that the deviant tones are louder and unexpected. So in the N1, you can see that because they're louder, they make a larger N100. That's auditory sensation. We increase the loudness, bigger component. And because it was unexpected and it was a deviant, they create an unexpected, aha, what was that? The stick snapped in the woods. That's why you see the separation between the standard and the deviant. So now I'm training your eye to look also at the difference between the two conditions. And to see the difference, you can actually now take a look at it in microvolts and assess for yourself, is that a significant difference? And remember, I told you, anything greater than 1 microvolt is a significant difference. And so these responses are about 6 microvolts in the negative direction and 16 or 14 in the positive. And the difference even between the conditions is greater than 1 microvolt. So it's definitely a significant response you're looking at. And now we can look at the individuals in the N400. And again, look at the top right-hand waveform. Here, you see very clearly 2 bumps in this peak. We call this a bifurcated peak. In both, you see that it's much broader, and that's because it's reflecting a complex underlying process. But here, you can also see that you can look for the separation between the incongruent and the congruent. So you're really trying to look for airspace between these two. And when you see this airspace, again, in the N400, it's not always large, but it's always important. So the size is not what you're going for here, but the directionality definitely is something your eye is going to learn and train on to make sure the incongruent is showing larger than the congruent. So now if we can move to the next slide, please. Thank you. Now here's an interesting play on the same variant. I'm showing you now just the N1 and P300. But what I've done now is I'm showing you the N1 and P300 on the top row like you know it. This is for auditory tones. So this is the neurocatch sequence as you have it. The bottom is to demonstrate the point that these responses are multimodal. So we've published a paper, and this is from that paper, that shows what neurocatch looks like if we present it to you visually. So the first thing I want you to do is start back at just start at FZ or CZ. We're going to talk about electrodes in a minute, but start back at taking your eye through the waveforms. Notice the pre-stimulus baseline is flat. Notice in the auditory, the N1 is much larger, and then it goes to the P3 like you've seen it. Notice the smaller signature in the standard versus the deviant. Now, move to do the comparison of what happens when we change from giving you information as you hear it to giving information as you see it. First of all, we still get an N1, but now you actually get a very strong P2 component. So notice the N1 move to a P2 and then a P3. This component is not specific to visual, but in this demonstration, it really shows the P2 well. So you can see that you might almost consider this as like, is this one peak that's bifurcated? It's not, it's actually two separate peaks, and you can really see that clearly under the FZ electrode. And so the key thing here to recognize is this can actually also happen in individuals in the auditory modality. Some people might have two peaks, some have one. And the algorithm versus the trained eye is in its first version was breaking down on this, and it was sometimes it would bounce between them. If it bounced between them, you can imagine you'd have very different latency and amplitude scores, particularly latency. So the next generation, and the team has a fix for this that actually catches this. But I'm going to give you a metaphor in terms of how to think about that and why it's important that you know how to read these waveforms. Because sometimes it's still a very intact sensory processing, but what we're doing is we're training an algorithm to be as good as the human brain in pattern recognizing, and we're not going to get there in our first generation. Last thing on this slide, take a look at the N100 in the auditory condition from FZ, CZ, and PZ. So remember, I taught you the electrode nomenclature in terms of F being frontal, C central, and P parietal. So that's a front to back. And look at how the response actually reduces in amplitude. Compare that to the P300, which actually maintains or sustains in this individual in quite, and in fact in the visual, gets quite a lot larger in the PZ electrode site. So that's a demonstration of change over space, right? We can identify components also, not just by looking at their waveforms, but also by looking at their topography. And a prime example of that, which we've dealt with in the NeuroCatch platform that you don't have to worry about, are blinks. Your blinks make big artifacts. I think I mentioned it last time about the cornea being positively charged and causing, and we can identify a blink because it's largest in the front and it actually gets smaller as it goes towards the back of the head. So if I ever have to worry about artifacts, alpha would be another example. If I'm looking at alpha, I can expect it to be larger in the CZ, PZ, and it's not as large in the front. So that's where the space information is also useful. You don't have this built in NeuroCatch. We optimize for it. So for the components, we actually combine the electrodes to give you the best picture. And we also optimize for these artifacts. We take them out. However, let's pretend that it's not perfect. So in an example where we would worry, do we have alpha as an artifact? And that's the most common artifact that can happen, particularly if patients are sleepy. What would you do to look at any result to assess whether or not that was due to alpha versus a legitimate peak? Now, I'm not going to make this a sort of question and answer. I'll let you think about it. And while I'm talking, hopefully you come up with it. But the key thing to start first is you would see alpha in both conditions. So it will be present both in the deviant and the standard, and in the congruent and the incongruent. So if you see only one peak in one condition and the other condition doesn't show alpha activity, it's likely not alpha. Could I get the next slide, please, so we can move to the N400? Okay, so let's do this again. Oh, back up. And quickly go and show yourself, walk yourself through the N400. So start again, the auditory is the neurocatch sequence as you know it. And look here, you can see the P1, the N1, the P3 or P2, it's difficult to say in that. And then you can see the strong, broad peak of the N400. Now I put some extra letters in here to alert you to the fact that this is actually a component we discovered. I did my PhD on these two components called the PMN, the phonological mismatch negativity. So you can see quite clearly this preceding peak and then the larger. If you look in the PZ electrode or the CZ electrode, there's this sharp peak in front of the complex, and then where those two stars are is actually the N400. That's actually, we've separated that. That's you listening to it didn't match as it sounded, and then it doesn't match as it means. So that is why the N400 is a different shape from the P300 and the N100. And you can literally train your brain on what that looks like. And what's interesting is you can compare that to visual. So when we hear, we get information over time. And when we see or read, we get the information instantaneously. So look at the peak differences between the top and the bottom row. And the first thing you can see is that the information for the N400 peak in the visual is much sharper. That's very characteristic of a visual N400 because you've got the instantaneous information coming through the visual system, whereas when you're speaking and you're listening, you're getting that information over time. And you can see those differences in the waveforms there. The other little embedded Easter egg, if you will, in this slide is your inner scientist probably noted that you don't see a PMN. So you do not see the peak that is to the unexpected sound because you were reading. So it doesn't appear in these waveforms. Next slide, please. All right, so now let's move to understanding, okay, these are the waveforms as we record them from scalp electrodes. But what does that actually look like when we sort of dig into the brain, so to speak? So on the left, if we just look with the N100 with a bunch of electrodes, and this is an experimental result I'm showing you where they played it only to the left ear. So they were intentionally trying to show that it was going to the right auditory cortex because our brains are very crossed, as you know. And so what you can see is just like a peak in a topography map, the N100 is peaking over the frontal regions, but it's asymmetric towards the right and demonstrating a strong right asymmetry towards the temporal lobe. And on the right-hand side, when we take that information and we go and we look at the source, so we can actually use MEG as an example to go find the source of this N100, it localizes very neatly to the primary auditory cortex. And you can see it's in the right primary auditory cortex. So the N100 very much does reflect how your brain processes auditory sensory information from the time it hit your ear to the time it hit your primary auditory cortex. And there you are looking at it in the reel. So if we can move to the next slide, what does it look like for the P300? So again, let's go left to right and break it down. First, what I did is I selected some research that is really interesting because what you can see here is the P2 component, and then the fact that the P3A isn't just one component. It actually has subcomponents. And often with these, they do. Your brain is very complex and it does lots of things and we capture it in its electrical changes. And so a P3A is actually if it's a novel unexpected stimulus, and a P3B is if it's a targeted unexpected stimulus. And they've been demonstrated experimentally to be quite different. Why am I showing this? Well, number one, you can see the P2 is distinctively different than both P3s. And number two, we're really in neurocatch looking at a combination of the P3A and P3B. So they're melded together. But what you can do is take your eye through and see the baseline, pre-stimulus baseline's flat. Now you see the clear P1 and N1 and P2. Sorry, they flipped this. That's another thing I should tell you. What they've gone and done, and this happens sometimes, NeuroCatch flips it for users, but by convention in ERPs, historically they plotted negative as up and positive as down. You can blame electrical engineers because it was actually those guys that did it. But what's happening now more and more is people intuitively think positive should be up, so waveforms are being flipped. So I was talking too fast and I missed the fact that in this one, they've flipped the waveform so you're seeing the P3 up. This is actually good in terms of training your human brain because you can actually train your pattern recognizer by twisting the waveforms around and see if you can still see it. So let's walk through and you can see now we can see the early P1 dip and then the N1 and then the P2, which goes on to the P3. So hopefully you can kind of see that and you've been able to localize that. Now what you can do is actually move to look at the features of this in terms of the middle of the graph. So the P3A has a very central parietal topography, and the P3B in this case is much more posterior. So you can say, well, that's interesting and exciting, but that's just the sort of electrical activity on your scalp. Now, what does it look like in your brain? This is where we get to that lesson in functional anatomy that I mentioned, that information is both localized and distributed at the same time. That's the paradox. And so what you can see with the P3, which is a higher level process of attention, basic attentional processing, it is now not localized to one specific area, but rather this is showing you MEG results for the P3B and demonstrating that it is a distributed network of active areas in the cortex that are very much involving your parietal, your temporal, and your frontal lobes. And so a pretty critical thing to think about in this case, and it makes perfect sense in terms of attentional cognitive processing, it's using a lot of your cortex in a distributed way. Next slide, please. All right, so what about the N400? Well, let's start with the topography map. First of all, you're going to notice that this is a centrally parietally oriented N400. These are to word pairs, auditory word pairs. And this is actually our own data on the right, localizing the N400 for the NeuroCatch sequence. And what you can see is that when we actually look at activity that's the incongruent greater than the congruent, in the left hemisphere, it very much matches our classical understanding of the organization of language. So we have both the Wernicke's and the sort of posterior temporal and parietal. We have strong temporal lobe activation, but we also have inferior frontal for expressive and rehearsal, and dorsolateral prefrontal cortex involving verbal working memory. If your eye looks between the left and the right hemispheres, you can see that while it is larger in the left hemisphere, it is still absolutely the case that language is distributed bilaterally and also includes what we call homologs in your right hemisphere. And then your sanity check is when we reverse those conditions, all that activity drops out. So you see nothing. So the N400, again, distributedly localized, but very much consistent with your cognitive, semantic, and language networks. Next slide, please. All right, so let's walk through some interesting stuff about what these components look like. And I was picking a slide for each. So this is the N100 in auditory neuropathy patients. On the left is latency, on the right is amplitude. The black is your normal controls in a quiet and noisy environment. And the blue are your auditory neuropathy patients. So start with your normal in a quiet environment. Those are the black filled dots. And what you can see is that on the horizontal axis, this is decibels in terms of sound level. And as you increase the sound level, you get a faster N100. So there is a point where if the sound is low, the latency of the N100 slows. And when you add ambient noise in, you see a very distinctive change in the latency of the N100 in terms of now only when it's loud enough do you get the same latency levels. When you compare that to individuals with auditory neuropathy, the pattern is quite telling because now, of course, auditory neuropathy looks like more or less the same as individuals in a noisy environment. And then that effect gets exaggerated when you move them into a noisy environment. So that's a really nice graph to give you a sense of the timing of the N100 in terms of how it responds to manipulations of sound and also individuals with or without hearing challenges. The amplitude is much the same on the right-hand side. And you can see they've put little cartoons in here to show you that the larger the amplitude, with the larger the sound. And you've seen that in your own NeuroCatch data that we presented. But also you can see the same pattern where when you add noise, that has an impact on the amplitude, less so than the latency. But that impact equals roughly the same as somebody with an auditory neuropathy. And then that gets exaggerated when you add noise with somebody who has an impairment. I like that slide. I hope that captures effectively the way to think from a functional point of view of what changes in the N100 are really telling you. There are many other sort of nuances to it, but I think that captures the basic gist. Next slide, please. All right, so what about the P300? I apologize, I worked really hard to get better graphs. These were the best I could get. This is actually off of Wikipedia, although I've got the source down below for you if you'd like to grab it. But I think this is very important characteristic curves to keep in the front of your mind. Again, latency is on the left, amplitude's on the right, and we're looking as a function of age for P300 latency and P300 amplitude. And they have both incredibly characteristic age-related curves. And what you can see is you can get a sense of not only the amount of variance in these norms, but also the age-related changes as we go from being sort of in our peak low latencies and high amplitudes in our sort of early 20s, let's say, range, to characteristically slowing and getting smaller with age. And you can see that also an element of the variance. So if you look at the upper and lower bounds of both of these, you can get a sense that the data we've given you on the onboarding in terms of the latencies and the amplitudes are basically captured in this snapshot. So that's a really nice normative thing. And again, I'll reinforce for the P300, you're really looking at the P300 in terms of its measure of your information processing. And the key thing about that is it's like blood pressure with white coat syndrome. If it can be variable. So individuals can vary, particularly depending on their current state when you're seeing them. If they're well rested, they've had a good sleep, and they're feeling awake and alert, they will give you a very good P300. And if they were out partying the night before and haven't had coffee, it will change. In our labs for our students, they do experiments where they do pre and post caffeine, and the latency of the P300 changes. So please be aware that while there are norms, this component particularly is very specific to how the individual is on that day. Next slide, please. This one is less so. So N400, I'm showing you the same sort of thing. Age is on the horizontal axis, and you're looking at latency and amplitude related changes. And here, what I really want to drive home is that there is age-related effects with the N400 too. So if we're looking at people that are into their 70s and 80s, it is not uncommon to see the latency not be right at 400, but actually be sort of more towards the 500 millisecond range, and to see the amplitude actually start to be around 2 microvolts. Whereas if you contrast that with looking at somebody in their 50s as an example or younger, then they can have peak latencies that are well below and faster than 400 milliseconds. And they can have amplitudes that are on the order of 7 microvolts large. And notice, of course, that the N400 is a negative going peak. So that's why, in this case, going down towards 7 is a good thing, not a bad thing. So that just gives you a sense of the N400 as a function of age. Next slide, please. All right, so let's put it all together. This is also results that I showed you on your onboarding related to our concussion work with Mayo Clinic. So these are hockey players that had acute concussion tested right off the ice. And we got them both at their baseline, in a matched baseline when they were training, which is in blue, at post-concussion and at return to play, when they were cleared to return to play. And so let's start with our ability to read waveforms. So here we're checking the baseline. It looks good. You can see the P1. It goes into the N1. You can see that the N1 from baseline to post-concussion increases in amplitude and delays in latency. And then we watch that move into the P3, where again, it increases in amplitude and delays in latency. Then we can compare that if we stay on the top row and move to the right. Hang on, nope, pardon me. Go to the bottom row, I directed you wrong. Go to the bottom row, 3 lines. The N400 from baseline increases in amplitude and delays in latency. And then what's interesting is you're probably now looking at the green line, which is the return to play. And this is when the clinical decision was made to return these athletes to play, because based on behavioral paper and pencil testing and everything else they had, they thought they were recovered. And here you can see that for the most part, that's actually pretty accurate. The N1 is pretty much coming back to where it was. The P3, same. The N400 is also back in latency with a larger amplitude. That may be due to the fact that the N400 amplitude can vary within individuals. But one thing that was significantly detected at the group level is actually the P3 amplitude was significantly altered still. And so there was one measure within all this that detected that they hadn't gone all the way back. Now on the right side, this is subconcussive results. This is just another way to train your brain. These are athletes that didn't have concussions, no diagnosis of concussion. And your blue line is your preseason and your red line is your postseason. And here you can see that they match up very nicely in their N1 and their P3, but there's a significant delay in their N4 latency. Now, what's interesting here is now it's becoming kind of expert peak pickers. You can see that the latency delay, the N400, which is broad and has this preceding PMN peak, looks to be in there, but the semantic processing is what was delayed. So the players changed in terms of their reliance on the sound expectation in the early season, and then their reliance on the meaning expectation. And that significantly shifted the overall peak in time. And we're just about to publish, and we've since replicated this in Bantam and juniors, and also shown that Bantams who have less hits have far less effects, and junior A's have far more effects. So that's an example of how that looks. And if we can switch to the next slide, I think you get to see this in radar plots. So here's mapping all 6 on a radar plot. And here's your baseline, this is version 2.0 coming your way in 2020. So now your eye is saying, okay, that's great. I'm an expert at reading the waveforms, but I really like the hexagon because I know that's just a simple way to represent this in a normative framework. And all 3 peaks, their amplitudes and latencies, so 6 measures are shown. And if you have a proper within norm response, you get a fairly symmetric hexagon around the 50% mark. And in this case, because the players, their amplitudes all went up and their latencies all delayed, that shifted the hexagon pattern characteristically into a triangular pattern. And then what you can see is when they were returned to play, it was shifting back to the hexagon pattern, but it didn't shift all the way back and was still significantly out. And then the far right just shows you the pre-post difference in terms of the subconcussive impacts for players that didn't have a concussion. So that's sort of the next generation of what's coming, and that is the end of module 2. How do I do for time? 42, left some questions. Time for questions.
I have a question.
Mm-hmm.
Well, when you were saying about the N400 that athletes, when they were pre-season and post-season, the graph, so that they have latency slower, is that right? Or the latency increased?
Yes, that's correct.
That's correct. Would it be because you are saying that it might be because, how many times they were hit on the head or, things like that, but would it not as well after the post-season as well, because they are trained mentally as well, maybe just focus on the game, not to focus, not to pay attention for the words, or just, shut off everything else. So, you know what I mean, that they are mentally trained for, just focus on something else, right, on the game.
Great question. There's much debate in the literature about the N400 in terms of the concepts of, is it automated, like automatic processing, or how much do you have kind of attentional and or volitional control over it? The short answer is, without getting into kind of the conceptual arguments, it's very automated, right? So if I want to get an N400 in you, I just have to say the pizza's too hot to sing. And I got an N400 in you. And if you were a hockey player, whether or not you were at the beginning of the season or the end of the season, I would get the same N400, because your brain has this machinery where, as I'm speaking, it's actually skipping ahead to anticipate where I'm going. You're filling in the words before I say them. And so that's the trick, is that if I set up your brain to sort of get ready, and then I violate, your brain goes, that didn't fit. Now, that being said, and just so you know, it's incredibly robust, right? We started working on and discovered the N400 in 1985, and it's been open sourced in the research community since that time. And there's lots and lots of literature. It's incredible, actually, as a component. You can get it in the blind in Braille. So our brains have these meaning traps that they're sort of, the information is coming in, we're processing it, and if it doesn't match, this thing happens, no matter how it comes in. And so the key thing is that, of course, like anything, you can still, and it has been characterized quite well, influence that system. So habituation is a particularly something we are well aware of and quantified and have control. So if I give you the pizza is too hot to sing again, this time it wasn't as unexpected. And so if you were to do the neurocatch sequence in any individual, we've done it in hockey players as an example, and others, within the same week, even though the stimuli will change, there will be a characteristic drop in the amplitude of the response, because people figure out that, oh yeah, well, this is where some words match and some don't. And so the level will change, but it won't drop below the level of significant. It will only drop, I think on average, it might go down a microvolt. So if it was 4, it'll be 3, that sort of thing. So you would expect a characteristic habituation if you gave it lots of times, but you would never, so you wouldn't worry too much about that, but you wouldn't expect a change in the latency, in the timing, because that's really important. That's the time in which your brain is accessing information. So if you see a change in the timing of that, that would reflect that something has if it's significantly changed, in this case as it did, it reflects that somehow they were not processing information as fast as they used to be. And it's a little bit more complex in the waveform, and I pointed that out in terms of that it's a bifurcated peak with this phonology and the semantics. And so there was a shift in that where there was an emphasis, but ultimately that was still an overall change in the way they were processing and cognitively processing information.
Okay, thank you.
Great questions. Yeah, these are good. Keep them coming. The way to train yourself to read these things is very much to ask these questions, dig into it, and see the waveforms over and over again, like what I tried to do. The other thing to be aware of, I actually take full responsibility for this because I only just realized this and I missed this. In NeuroCatch 1.0, we made it inadvertently harder to read the waveforms because they basically take a window. They don't take the full minus 100 to 1,000 millisecond. They only give you the snippet. And then of course, those graphs don't even put time or amplitude on. And so the reports as they are right now are definitely going to be improved upon. So I actually have to accomplish training you better because I, even when I see them, don't have all of the information that I necessarily need. And that was a definite area we missed when we rolled out the first report. I missed.
So can I ask a question about that then? Is that the same thing? Because the N100 and the P300 are not on the same graph. So this we're on the same graph this time. So now, is that a change as well?
It will be, yeah. We're going through the new report now, which will incorporate both hexagons and improved graphs. And I think what happened, and I missed it, but they basically took the window and should display the N100 for its window and display the P300. It was kind of engineering thinking of, okay, here's the N100 in its window, here's the P3. They're actually taken from the same waveform.
Okay.
Mm-hmm.
I have to just say, because I don't know if you can see this, this is what I'm used to. So I was a bit in cognitive dissonance the whole time looking at it when you're explaining them, because I'm used to seeing them separate. And not that I've even done that much, but in my mind, because it's relatively new material, I've just compartmentalized it. So I must have an engineer mind, even though I'm a psychologist.
Well, it's also the way that we've taught you, and that's okay. Hindsight's 2020. I think I think at the time we were really trying to just allow the peak picking to go and give you the waveforms and not have to necessarily do any human pattern recognition. But what in the third module, what you'll see I'm going to be talking about is two things. One is we're going to use those reports. So I'm going to show some results, and then we're going to select reports and we're going to go through them. And I think everyone really enjoys the third module, because then you can translate from understanding the waveforms into reading. So we'll try and help there. The second thing is that I've come to really understand, and I'll talk about this and I'll show this, but the metaphor of automated driving is very much in play in terms of the development here. And of course, the engineers have created 5 levels of automation, and it applies to the peak picker as well. So in the first generation with early adopters, we're trying to better support you all in terms of, like my car has the ability to see like many of the lines on the road, and that's like a level 1, level 2 automation, but I still have to keep my hands on the wheels, because if it's snow covered or otherwise, right, I still have to do the driving. And I think we're probably, I'll show you the graph in the next module, but I think we're probably about 2, 3 on that step. And the next generations are getting better and better. So they will remove the need to be able to just see the waveforms. One of the big goals is to help you translate between the report and what they actually look like in real life.
Okay. And the hexagons, if you're in the space that we would be using it, I love that, because we're using it in more of a healthy kind of aging wellness space, because the interpretation, I think, is way more clinical for what we would use it for. So those hexagons, love it.
Yes. Yeah, we very much love the hexagon. It's been a goal to get away from the squiggly lines, which make an access barrier. But part of the Electrophysiology Bootcamp is just recognizing that as fast as we can get away from them, we still have awesome users like you, all of yourselves, who we have to support better, so that until we're there, they don't just look like you're looking at tea leaves in a teacup.
Yeah, for sure.
Yeah. More questions. These are all very good.
Well, as I've said to you before, Ryan, being primarily a clinician, my interest is how does this correlate with how a person is doing clinically and functionally in their life. So that's the patient, that's what they're after, is their function improving. And that really is my interest in being able to interpret these. How do you correlate these squiggly lines with what we're observing in our interactions with these?
For sure, John. And I wrote that down, and I thought you captured nicely the sentiment that we get quite commonly from our clinical early adopters. And the key short answer of that we've found in our clinic, and that I'm really trying to help provide support in terms of training here, is to be able to look at the squiggly line to know, all right, it's not an artifact. And we saw a data set from you where all 3 responses were below 1 microvolt, which right away told me that the reference electrode was off. So just like in radiology, the first thing I do when I see an MRI is I look and I say, if like I'm testing and I see something, is this an MR artifact or is this something I need to pay attention to? So, so the ability to know the responses are there. Then the key thing for patients and how does it correlate clinically, in my mind, is very much the first and most important thing is the timing of how your brain is processing information. So I immediately look to, okay, if I've got good responses, are they the same, which is great, they haven't changed. Are they getting faster? If so, is that because I would expect that because I had somebody come in with mild traumatic brain injury and I've been doing a PONS protocol and they are reporting that they're getting, their cognitive processing is getting faster. And if that's the case, do I see that in the actual millisecond measurement of how these responses are? And then finally, I look back at amplitude for interesting clinical things. And we use the example of a PONS patient who had auditory sensitivity and came in with earmuffs. And then as treatment and therapy was improving that, the N100 amplitude dropped down. And so understanding that the amplitude reflects how many cells in your cortex are firing in synchrony means that you can put in a correlation to, does that make sense to in what I'm seeing as well? And they will, just like blood pressure metric, they will change to some degree. They'll vary a little bit, and then then there will be changes that will very much, you'll be like, I think that is really correlating clinically. And we do, I can tell you, in looking through the different reports, we are seeing quite a lot of encouraging results in that line.
Are there questionnaires that you use with people that you could ask, in particular, are you more sensitive to noise or light, or like, are there standard questionnaires that you would ask, not around the concussion space, but just if somebody's feeling like, oh, I'm quite foggy and I don't feel like I'm as with it. Like, I think even I tested, because we're still we haven't sold any, to be fair, but we're still testing people. And because I'm perimenopausal, I'm testing lots of perimenopausal women, and everyone feels like their brain is in a fog. And then, but how do you compare that? Like, can do this scan and say, okay, it's normal, but.
Yeah, so that's a great. So first, the first thing you're going to want to do is look at it based on the package and the information we've given to say, okay, these are all sitting, I see the responses, they're normal, that's great. So everything is in normal range. Within our clinic, we have certain things we've run. In the paper that I showed you, the 2016 paper, we actually did look at lifespan related effects and use things like the MoCA and the MMSE to measure change against change. And in general, what we find is we're much more sensitive than existing tests. So when you do make a comparison, sometimes you'll see where we detect something and the test didn't necessarily. And in that case, the older adult sample was, of course, fine with all those measures, and we detected a significant delay in the P300 and actually just near significance in the N400. So that's number one. Also, if somebody comes back subjectively and says, yeah, I'm not, I'm feeling less foggy, I'll sort of answer the question with a question. What is the first component that I would look to?
I'm sorry, for what? Oh, well, for fogginess?
Yeah.
Oh, I would ask sleep. Are you getting enough sleep? And like lifestyle, that's what I would ask. But
right, but of the three components, where would your mind go to first to take a look at? There's two in order one and order two.
You mean in terms of like the waves, like the P100, P300, or N400?
Yeah.
Is that what you mean?
Yeah.
Well, I don't know. The N400 is a function of age. I'd go to that one.
Right.
Yeah.
So if I thought it was a transient, I'm feeling better today, I'm less foggy, I would look to the P300 because that's much more how your information processing is today, affected by things like coffee or sleep or otherwise.
Which I have right here in my notes, just so you know.
And if they said, you know what, over time, some of my patients, they've reported, wow, my cognitive processing is better, then I would definitely, if it was more of a sort of, rather than a transient, but more of a kind of a consistent finding, then I would look to the N400 as well. So in order, my eye would go check the P300 and then check the N400. And I would look first at, assuming they were responses were in range, I would look at the latencies, the peak latencies. And second, I would look at shape.
Okay.
So I just want to be cognizant of time. We are kind of at the end here. Robin just messaged me saying, sorry, he had to step out, but he's looking forward to being on the next call for bootcamp number 3. And I will be following up with everyone for bootcamp 3 because we will be discussing the cases that you guys bring forward and that you have questions for Ryan to answer. So I'll compile them and kind of control, the screen like usual, and that way we can have them ready to go and make it through as much and as many as we can. So do we have any other questions before we kind of wrap it up or anything like that? Ryan? Nothing?
I do, but I'll ask next time, I think, because we have to.
Yeah, thanks very much. We have to go.
Yeah. Thanks everyone, and just bring forward your cases that are interesting so we can discuss them. The third session is very much a fun session.
And Marcella, if you have any questions, you can email me as well.
Thank you.
Yeah.
Thank you.
Take care, everyone.
Thanks everyone. Sorry for the muddy face.
Bye.
Bye-bye.
I've got to